The fluid mosaic model of biological membrane comprises of phospholipids and proteins that are constantly moving within the layer in a lateral motion. Proteins are interspersed in the phospholipid bilayer, giving it a mosaic appearance. A phospholipid is an amphipathic molecule, meaning it has both a hydrophilic region and a hydrophobic region. More on the significance of this will be elaborated later. Now, before jumping into the topic, let's study the history of the discovery of the fluid mosaic model. Scientists began studying membranes decades before membranes were first seen with the electron microscope. In 1915, membranes isolated from red blood cells were found to be composed of lipids and proteins by chemical analysis. So, how did they isolate the plasma membrane from red blood cells? Well, first you must have a blood sample and then centrifuge it. After centrifugation, decant the supernatant and you will be left with the red blood cells. Now, add some hypotonic buffer to break up the red blood cells before centrifuging it again. After a few more rounds of centrifugation, you should be able to distinguish the plasma membrane of the red blood cells clearly, as the red blood cell has no internal membranes and organelles. This is also the reason why scientists use the red blood cells in their studies initially. In 1925, 10 years after the initial studies of plasma membrane by scientists, two Dutch scientists reasoned that the cell membranes are phospholipid bilayers. So if phospholipids are the main fabric of a membrane, then where are the proteins? Though the heads of the phospholipid molecules are hydrophilic, the surface of the phospholipid bilayer adheres less strongly to the water than that of a biological membrane. Thus, it was proposed by Hugh Davison and James Daniell that the membrane is coated with hydrophilic proteins on both sides, and this is called a sandwich model, where a phospholipid bilayer is sandwiched between two layers of protein. Though in the 1950s, the study of membranes with the electron microscope seems to support the sandwich model, by the late 60s, problems with the model were revealed, and based on these observations, two scientists came up with the fluid mosaic model in 1972, and this is the model accepted today. Now, let us start with topic proper and zoom into the different components that make up a biological membrane. Firstly, let's start with phospholipids. Phospholipids have a hydrophilic phosphate head and two hydrophobic hydrocarbon tails. Thus, they are referred to as amphipathic, and their amphipathic nature will allow the phospholipid molecule to form bilayers in aqueous environments. The hydrophilic phosphate heads face the aqueous environment at each surface of the bilayer, while the hydrophobic hydrocarbon tails are shielded from the aqueous environment in the interior of the membrane, forming a hydrophobic barrier to polar and charged molecules. Transient gaps also exist between phospholipid molecules in membranes to allow certain small particles to pass through. Thus, phospholipid molecules play a big part in the selectively permeable nature of membranes. Now let's go on to membrane proteins. There are two major groups of membrane proteins. First are the integral proteins. They penetrate the hydrophobic core of the phospholipid bilayer. Many of the integral proteins are transmembrane proteins which span the whole width of the phospholipid bilayer, while the rest only extend partially into the hydrophobic core. Integral proteins are held in place by hydrophobic interactions between their hydrophobic regions and the hydrophobic hydrocarbon tails of the phospholipids. The hydrophilic portions will interact with the phosphate group of the phospholipids and the aqueous medium on either side of the membrane. Then there are the peripheral proteins, which are not embedded in the phospholipid bilayer and only loosely bound to the surface of the membrane. They tend to be rich in hydrophilic amino acid, which allows for interaction with the hydrophilic portion of the membrane and the surrounding aqueous environment. So, what are the functions of membrane proteins? Firstly, they can be utilized for transport. A protein that spans the membrane may provide a hydrophilic channel across the membrane that is selective for a particular solute. Other transport proteins shuttle a substance from one side to the other by changing shape. Some of these proteins hydrolyze ATP as an energy source to actively pump substances across the membrane. Membrane proteins are also involved in enzymatic activity. A protein built into the membrane may be an enzyme with its active site exposed to substances in the adjacent solution. In some cases, several enzymes in the membrane are organized as a team that carries out sequential steps of a metabolic pathway. A membrane protein may also have a binding site with a specific shape that fits the shape of a chemical messenger, such as a hormone. The external messenger may cause the protein to change shape, allowing it to relay the message to the inside of the cell, usually by binding to a cytoplasmic protein. This is called signal transduction. Some glycoproteins serve as identification tags that are specifically recognized by membrane proteins of other cells. This is called cell-cell recognition. Membrane proteins of adjacent cells may also hook together in various kinds of junctions, such as gap junctions or tight junctions. This is called intercellular joining. Finally, membrane proteins may be used for attachment to the cytoskeleton and the extracellular matrix. Microfilaments or other elements of the cytoskeleton may be non-covalently bound to membrane proteins, a function that helps maintain cell shape and stabilizes the location of certain membrane proteins. Proteins that can bind to extracellular matrix molecules can coordinate extracellular and intracellular changes. Now let's go on to membrane carbohydrates. Membrane carbohydrates are short branch chains fewer than 15 monosaccharide units. They are covalently bonded to proteins to form glycoprotein and covalently bonded to lipids to form glycolipids. Glycoproteins and glycolipids serve the same purpose of being cell surface markers for cell-cell recognition. They are involved in cell-cell adhesion and also serve as specific receptor sites for signal transduction. However, glycoproteins and glycolipids are also involved in protecting cells against mechanical damage and keep cells at a distance, preventing unwanted protein-protein interactions. Since a membrane is held together primarily by hydrophobic interactions, which are much weaker than covalent bonds, most of the phospholipid molecules and some of the membrane proteins can shift about laterally in the plane of the membrane. Flip-flopping transversely across the membrane, however, is rare since it will require the hydrophilic part of the molecule to cross the hydrophobic core of the membrane. The fluidity of membranes has to be precisely regulated. Certain membrane transport processes and enzyme activities cease when membrane viscosity exceeds a certain threshold level. The fluidity of a phospholipid bilayer depends on a few factors. Firstly, as temperature decreases, membrane fluidity decreases, and as temperature increases, membrane fluidity increases. The degree of saturation of hydrocarbon chains also affect membrane fluidity. Unsaturated fatty acid chains with cis carbon-carbon double bonds have kinks. These kinks hinder the hydrocarbon chains from packing closely together, thus increasing membrane fluidity. Saturated fatty acid chains are long and straight hydrocarbon chains. This facilitates close packing and thus decreases membrane fluidity. Next up, we have cholesterol. Cholesterol is a major component of membranes in animal cells, where it serves as the key regulator of membrane fluidity. Plant cells lack cholesterol, but contain related compounds, steroids, that fulfills a similar function. Cholesterol contains a hydrophilic OH group, but most of the molecule is hydrophobic. When present in membranes, cholesterol is commonly wedged between phospholipid molecules, with its OH group aligning with the phosphate heads of the phospholipids, and the remaining hydrophobic portion tucking into the hydrophobic core of the membrane. At higher temperatures, cholesterol limits fluidity. This is because its rigid steroid rings interact with and partly immobilize adjacent phospholipid molecules, thus restraining their mobility. This contributes to membrane stability at higher temperatures, and at lower temperatures, cholesterol interferes with the close packing of the hydrocarbon tails, therefore enhancing membrane fluidity. The presence of cholesterol limits the permeability of membranes to ions and small polar molecules. It does so by filling in spaces between hydrocarbon chains of phospholipids, thus making it more difficult for such solutes to pass through the bilayer. Finally, the length of hydrocarbon chains also affect membrane fluidity. Generally, the longer the fatty acid hydrocarbon chains, the higher the melting point of the membrane. This is because a longer chain length increases the surface area of contact, thus increasing the tendency of the hydrocarbon tails interacting with one another via van der Waals interactions. You have now come to the end of the video. Hope you enjoyed it and learned a lot. And don't forget to go play the membrane game.